Chapter 4, Inside a Cell. So today what we'll be discussing is actually looking at the cell a little bit more in detail, looking both on the outside of the cell, at the cell membrane, as well as inside the cell, looking at the cytoplasm, so the cytosol and all the different organelles that are within the cells. So the agenda for today will be to first look at how do we go about even studying the cell, the fluid mosaic model, which plays a big role with regards to that cell boundary. We can call it the skin of the cell. We'll look into that in a little bit more detail. And then different organelles, everything from the nucleus, as well as the different organelles that make up the endomembrane system, the organelles that help provide energy source for plants and animals, and then following up with the cytoskeleton, what gives the cells its different structure. So first, let's look at the study of cells. So cells are super important, right? They're the smallest unit, and they make up the basic unit of life. And so without cells, we wouldn't be alive. We wouldn't be living. And so how do we first observe cells? So the first person to kind of really observe cells was Robert Hooke. So back in 1665, Robert Hooke was looking at thin corks, and he was kind of observing them, and he noticed that these thin corks, this is one of his drawings right here, had these little tiny structure that kind of remind him of the Montessori. So he called them cells. And so he was the first one to kind of observe cells. Following up, Antoine van Leeuwenhoek was the first to kind of form a microscope. So he built his own microscope, and he was just very curious, right? Like other scientists, he was very curious, and he was kind of looking at all these different things, including just kind of looking under his own cells. So he was taking, for example, dental scraping, kind of looking under cells, and he was the first one to kind of notice these structures, these living organisms underneath the microscope. And he called them animalcules because they kind of remind him like little animals. He wasn't at that time sure what they were. And what he was actually observing was bacteria. So you can kind of imagine that this is a while ago when dental hygiene wasn't as frequent. Not that we don't have bacteria in our mouth now, we do too, but that was kind of he was observing. So he probably had a lot of bacteria to look at. So he called these animalcule beastie. And then as time progressed, microscopes got more and more powerful. And so by the 1950s, we were able to use microscopes instead of using light. So if you recall in lab, we're using these microscopes that shine light through the specimen. But instead of using light, these scientists use electrons. And they found that when they use electrons, they can actually observe the different structures inside the cells. And that's when we were finally able to start observing organelles. And so going back to the discovery of cells, we have two scientists here in the 1800s. We have Thomas Schwann as well as Matthias Jacob Schlechten, who were observing again different cells, and they looked at different plants as well as animal cells and started proposing these different concepts of the cell theory, along with Rudolf Virchow, who actually, while observing under a microscope cells, actually saw cells dividing and producing two daughter cells. And so he, across the three and many other scientists, they proposed cell theory. So what cell theory is the theory that all organism consists of one or more cells. So if you remember, we talked about bacteria and how bacteria are single cell versus us, animals, we're multicellular, so we're made of many cells. And as we mentioned before, it was observed that cells arise from the division of another cell. So a pre-existing cell will divide and form two new cells. And as we mentioned before, cell is the basic unit of life. So it's structural organism. In an organism, it's kind of what gives an organism life, right? These cells are the smallest units of life. And then for us, who are multicellular, we're made of many different cells. Oops, sorry. And so what are some of the characteristics of cells? Well, All cells have plasma membrane, so they all have also known as a cell membrane, like this phospholipid layer that kind of surrounds the cells and actually form a physical boundary between the cell and the environment the cell lives in, right? And so you can see right here that when we're looking at, let me get my pen on. We're looking at this section right here. This would be the plasma membrane right here. So we can think of it kind of as the skin of the cell. Additionally, all cells have cytoplasm. So cytoplasm is all the material, where's my pen? Sorry. All the material that's inside the cell. So inside the cell. So we have everything from the cytosol, which is, we can kind of point that to just this yellow shade here. All the different watery substance that's holding all of these different structures together. So the cytoplasm is made of cytosol and all these different structures. So that's everything from the different cytoskeleton to the different organelles. So we'll kind of look at these organelles later, but all of these are in the cytoplasm. And then lastly, all cells have DNA. So in eukaryotic cell, the DNA is enclosed in an internal structure, which we call the nucleus, and we'll look at that in a little more detail later. And in prokaryotic cells, they also have DNA, but their DNA is not in the nucleus. It's not in the internal structure. So their DNA tends to be free form and inside it, and it tends to be circular. So a lot of times we'll call their DNA plasmid, but it's still DNA. So let's we look at the two main types of cell, which we mentioned, right? The eukaryotic cell, which is right here. And what distinguishes a eukaryotic cell is it has a nucleus where we store DNA, as well as organelles. A prokaryotic cell, right here, it does not have nucleus nor organelles. And as I mentioned, its DNA is a circular DNA that's in a nucleoid region, but it's not actually inside an internal structure, similar to like what we see in eukaryotic. Okay, so first let's kind of look at prokaryotic cell a little bit more detail. So prokaryotic cell are different organisms that are in the domain. If you remember when we talked about domain bacteria and archaea. And so both of these organisms have DNA, as we mentioned, but their DNA is kind of free floating. So you can kind of see right here, they're kind of free floating DNA. And they're smaller and simpler in structure. So what do we mean by simpler? That means that they don't have organelles like the way we see. They don't have this hierarchy of many different organelles encapsulating and separating the different function. They do have ribosomes, and ribosomes are the site of protein synthesis. So they do have ribosomes that synthesize protein, and they do have an outside layer that is also the plasma membrane. So we can see right here, we're pointing to this plasma membrane right here. So the plasma membrane, again, is kind of like the skin of the cell. So it's kind of going to keep the cell's interior and exterior separate. And they also have, bacteria have something interesting called a cell wall, which is just a structure that provides the cell a little bit more support. The other type of cell that we talked about is a eukaryotic cell. So eukaryotic cells and eukaryotic cells are found in plants and animal and protista and fungi. So all of these are different type of eukaryotics, right? And a eukaryotic, as we mentioned, what distinguishes them is they have an actual nucleus. So we can see the nucleus here, as well as in this illustration, this right here is the nucleus. Additionally, a eukaryotic cell, has a lot of different organelles that help with its metabolism. So here we have these two are organelles. This right here you might recognize, it looks like a mitochondria. And then we'll talk about the endomembrane system, which is all this right here. And so these are different organelle structures that we'll go into a little bit more. And each one of these structures has a specialized function that it does within the cell. Okay, so here's another picture of a cell. And so this one right here, the first one we're going to look at is a plant cells. And some of the structure that's is different between plant cell versus animal cell is that a plant cell actually has a cell wall. So this provides structure to the cell. A plant cell, as well as different certain protein cells, have chloroplast. And so this is what's going to allow the plant cell to make energy using the sun. So it's going to convert the sun energy into a chemical energy that later will store as like starch. It also has a vacuole. So the vacuole will increase the cell surface area, and it stores in there sometimes water and also metabolic waste. So this is our vacuole right here. We also can find vacuoles in animal cells too, but we can really observe them in plant cells. So the next time that we actually look at the different plant cells within the lab, take a special note and kind of take a look at it, because you can actually really visually see the vacuoles in plants since they play such a strong function with regards to storing water in different plant leaves cells. In animal cells, you can see we don't have a cell wall. We do have the cell membrane, right? So the cell membrane is still this outer layer here, which we can kind of see that we're separating here. So this is cell membrane or plasma membrane. So we still have that. And rather than relying on a chloroplast for energy, an animal cell tends to rely on a mitochondria to help convert the glucose into different ATP molecules that the cell can then use as energy. There's a lot of other organelles, which we'll look at in a little bit. Okay, so let's move on to cell membrane and a fluid mosaic model. So the cell membrane, as we say, is the outer layer of the cells, the skin of the cell. And we're going to kind of look into that in a little more detail. So the basic structure of the cell membrane is actually a lipid bilayer. So if you recall, when we talked about the different macromolecules, we talked about lipids, and particularly we talked about phospholipids. So if you recall, a phospholipid has a polar head, and that's due to the phosphate and oxygen in the phospholipid, and it also has fatty acid. And so the fatty acid tail right here is nonpolar, so I'll just say NP for nonpolar, whereas the head is polar. And so the cell membrane is continuous and is selectively permeable. What I mean by that is not, even though it's a layer, it serves as a boundary because it doesn't let just anything go back and forth between the two. So small certain polar molecules can actually go back and forth between the outside cells. So we'll say, since there's cytoskeleton here, this is probably the inside right here, whereas this is the outside of the cell. So this is the environment of the cell. So not everything can flow in and out of the cell, but if we have small polar molecules, we can actually go in and out of the cell, between cell membrane. But for the most part, it allows us to have this barrier. You can also see that we have other things beside the phospholipid. We have cholesterol, which is here, and cholesterol plays a function with regards to structure and fluidity of the cell membrane. We also have protein. So this is an example of a protein right here. And this is also a protein, but it's called glycoprotein because it has sugar. So you can kind of see that these are ring structure, similar to what we expect to see of like glucose and fructose, these ring structures. So they're sugar molecules that are added on. And these sugar molecules actually, these glycoprotein that we see, these sugar molecules kind of helps differentiate different type of cell types. So they kind of function as almost like tag. They're like different tags that kind of tags and let other cells know that this is either a fibroblast or skin cell, or if they're a cardiac cell, and so on. So as we mentioned, the phospholipid molecule, as we talked about, has two different parts, right? It has the hydrophilic, so because it enjoys, or it doesn't mind interacting with water. So the water is on this side, on the outside cell, as well as inside the cytoplasm has cytosol, which is a watery substance. So the heads are facing outward so they can actually interact with the water, whereas the tails, the fatty acid tails, are hydrophobic. So they're scared of water. So they tend to be inside the phospholipid bilayer because they're kind of hiding away from the water. And so that's what forms this plasma membrane, is this double layer of phospholipid heads. And so we can call this the phospholipid bilayer. And so we're basically taking this part right here and zooming in to show you right here. And we kind of already mentioned the other different molecules that we find within this cell membrane. So why do we call this the fluid mosaic model? I think we have.
Between the living machinery of the inner cell and the harsh conditions of the outside world.
Okay, so the fluid mosaic model, and similar to before, all the links I will put in a different sheet so we can take a look at all the different, so you can take all the different videos. So the fluid mosaic model describes how the cell membrane is organized. And it's because we saw like the different proteins, right? So if you recall, we have, I'm not going to draw the full layers because it's kind of hard to draw with this pen. But if you recall, we have the phospholipids, and then we also have these different proteins as well as glycoprotein. So this is just my carbohydrate structure you can see here, right? So we have these protein and these glycoprotein between these two layers of phospholipids right here. And so it's fluid because this protein and this glycoprotein has to kind of migrate around and be able to have different functions, right? Because they actually have function. So these proteins can function as a way to transport things, because we said this is a barrier that doesn't allow things from the outside to go inside the cell. So these proteins can actually function as a way to help transport certain larger and non-polar material in, as well as water, in order for us to actually, the cells to rapidly move water in and out of a cell. We can actually use these different protein structures. So for water specifically, it's called aquaporin. But there are these different, they basically serve as a channel, right? And then these proteins, glycoprotein that we talk about, can serve as signaling. So sometimes you need to talk to other cells. So how do you go about that? We can give ourselves the different signals so that cells know what we are. And so if you want to interact with a cell, let's say we have a cell here and a cell over here, and they kind of want to be able to kind of identify each other with one another and show, then we want to move the glycoprotein that's over here to maybe move it to closer to over here. So this glycoprotein can actually move around within the cell membrane because the cell membrane is fluid. So they can kind of move around like a fluid. As we mentioned, these proteins have many different functions, right? So depending on what function it needs, we might need to move the protein around to different sections of the cells. And so that's why we want that fluidity. So proteins can function as channels. So as you mentioned, like an aquaporin is a channel, and it allows water to kind of move in and out. We can actively, meaning that we use energy or another cell or another molecule to actively transport cell. We can use it for cell recognition. So that would be, for example, the glycoprotein, where it has the sugar. We can actually use this receptor, so it can actually function. We'll draw a different protein here. It can actually function to actually recognize and bind onto different molecules. So that would be a receptor. These proteins can also function as enzymes. We kind of discussed when we looked at protein, how protein can have different function as enzyme. And then we can actually have these proteins function as junctions. So actually allowing us to kind of connect with other cells. Okay, we'll stop here. We'll move on to nucleus in the next section.